What do you really know about malaria? Hi, my name is Brian Bishop and I'm an undergrad researcher at the University of Notre Dame with the summer 2016 DISC REU. For more information, I'm going to put some links in the description if you want to take a look. I currently study at the University of Rhode Island as a PharmD candidate as well as a computer science double major. With this interdisciplinary background, I've been able to bring a really interesting approach to my summer research here regarding malaria and the drugs we use to treat it. Malaria is an infection that is caused by the Plasmodium parasite. Did you know it affects 20 million people every year and kills nearly 1 million of that 20 million? There are numerous drugs that we use to treat it, but each and every attempt has been foiled by the fast mutation rate of the Plasmodium parasite and some of the incorrect uses of the drugs. Over the past summer, I have taken a deeper look into the artemisinin drug resistance that is spreading throughout Southeast Asia recently. Specifically, I have been looking at the genome of the parasite Plasmodium falciparum. In this effort, we investigated and developed new methods for identifying copy number variations, or CNVs for short, in the Plasmodium falciparum genome. A copy number variation is just a fancy word for when a gene copies itself more times than it is expected. We use next-gen sequencing data to analyze two drug-selected Plasmodium falciparum genomes relative to their parent strain. When a genome is sequenced with a new type of sequencer, the genome is copied numerous times and then broken up into very, very small pieces. And then each piece is selected for a specific length using sound waves. So essentially, you have a picture of a puzzle, then you split it up into its individual pieces, and then once you have those pieces, you want to know what they look like, so you throw them through a sequencer and then you get the picture. Puzzle pieces are also known as reads, and once you have all the reads, you want to map them to an already known genome or put the puzzle together. And using the puzzle analogy, it's kind of like having a picture on the box to put all the pieces where they need to go. And since you copy the genome numerous times with the sequencer, you're going to have a lot of overlap with a lot of the positions. And just for an example here, at nucleotide base 15 you have a read depth of 5 because it's been shown 5 times. And at base 16 it was shown 7 times, so that I'd have a read depth of 7. We use the assumption that if the parasite has a lot of nucleotides in a row that have a high read depth, then it's likely to be a CNV candidate. Just to make sure that the CNVs were unique to the drug-resistant strand, we compare all the read depth values to the parent or drug-sensitive strain and then only took the values that were negative. We made the arbitrary cutoff at -50 just so we could say that this is interesting and it's not close enough to the parent strain. We then used the well-known theory of a hidden Markov model to predict the areas that have high potential to be a CNV candidate. The results found more than 243 CNVs on chromosome 5 alone. When compared to already known CNV finding programs where we were only able to find 2. Now take for granted there was a lot of overlap between that 2 and our 243, but our program was able to find CNVs at a variable length and found some that were not even found in the other program. This new approach shows promise in furthering our knowledge of malaria and equips researchers with new methods for analyzing the genome of Plasmodium falciparum. These new approaches analyze genomic data in a brand new way and shows great promise in identifying potential causes of drug resistance in the Plasmodium parasite. This is no means a one-solution-fits-all kind of solution, but successfully proves that the concept of this kind of analysis can be useful for finding CNVs. Thank you.